everybody, welcome to another episode of the Molecular Cell Biology YouTube channel. Today we are going to be introducing you to the idea of protein function. We are specifically going to address what dictates protein function and what are the key determinants that indicate what is any given protein going to be good for. Alright, so let's start with a very simple idea, and that is a protein is simply a consecutive sequence of amino acids. That's what a protein is all about. So from that point of view, we can define the structure of a protein, also known as the primary structure of a protein, as the specific amino acid sequence that forms that protein. So let's, for instance, say you have a protein that is about 92 amino acids in length. So this specific sequence of amino acids that make that specific protein is the primary structure of the protein. So let's just make it up. Let's make something, at least a few amino acids. Let's not go through 92 amino acids altogether. Let's start with, say, methionine, glycine, alanine, leucine, alanine, tyrosine, cysteine. Let's put another methionine here, blah, 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 blah. And let's say that toward the end, you have a threonine, alanine, leucine, and then maybe you have a tyrosine, aspartic acid, you have a lysine, arginine, and a glycine. Double glycine, let's make it at the end. That's the carboxyl end of the protein. And by the way, the other end, the very first amino acid, is the amino terminus of the protein. That's a 3, that's a plus. So amino to carboxyl, that's the way in which we typically refer to proteins, by the way, because this is the direction in which proteins are synthesized. They are synthesized from the amino to the carboxyl end of the protein. Okay, so the specific sequence, that sequence of amino acids that makes up this particular protein, that's the primary structure of the protein. Now, the secondary structure of the protein will be the specific shapes that will be adopted by specific subregions within the protein. So let's assume that our protein is now a spaghetti-shaped structure, and let's say the first section of the protein is kind of irregular, but it's followed by kind of a coil section, and it's followed by something that looks like a extended sheet that moves along, then connecting with another helical structure, followed by another final helical structure that goes to the other end, ending at the C-terminus. So each one of these subsections has very specific structures. For instance, this particular section here has an alpha helix. This other structure underneath here has a beta sheath structure. So this is alpha helix, that's a beta sheath. Those are the secondary structures of the protein. And the tertiary structure is the overall shape of the protein as a whole. So the totality of this drawing, the sum of all the regions that form the protein all together, that's what we refer to as the tertiary structure of the protein. Now, if this protein were to actually form complexes with another protein, so let's overlap now this image with that of another protein. So let's just put now another protein interacting tightly with this one. The sum of the two of them will then form what we will refer to as a quaternary structure. Quaternary structure. All right, so now, why am I talking about all of this? This is important because the sum of all of these components, the quaternary structure, in fact, of a protein, the shape of a protein, is what dictates the function, that the protein will have. And the function of the protein, being directly dictated by the shape of the protein, comes down to be dictated by the primary structure of the protein, meaning the amino acid sequence of the protein dictates in the end the function of the protein. And this is one of those absolutely essential ideas in molecular cell biology, that function is dictated directly by shapes, and shapes are dictated fairly directly by the amino acid sequence that forms the protein that we are referring to. Now, the function then is a direct consequence of the shape, but in order for a protein to provide the proper function that it is supposed to play in the cell, the other important component is the actual localization of the protein in the cell. So that's the other component that will be absolutely critical in dictating the function of the protein, the location that the protein actually has within the cell, cellular location of the protein. So that's going to be the second important characteristic that is absolutely essential in dictating functionality for proteins. And what dictates the location? What indicates where in the cell should the protein be located? Well, funny enough, it actually comes down to be exactly the same thing that dictates the shape, the amino acid sequence of the protein. Because in the primary structure of the protein, in the amino acid sequence of the protein, you actually find the key elements that indicate where in the cell the protein should be located. For instance, there are proteins that contain nuclear localization signals, which then make the protein be targeted to the nucleus. There are other proteins that contain targeting signals that allow them to get inserted into the endoplasmic reticulum when they are being translated initially. So there are sequences that will play their function early during the synthesis of the protein, meaning the targeting of the protein will be done co-translationally. As the protein is being translated, the protein will be targeted to the final location that it needs to be going in the cell, whereas there are other sequences, such as the nuclear localization signal, that play a role post-translationally, meaning once the protein has been fully synthesized, those sequences then are able to target the protein to the right location in the cell. So with this, I think that we have given you already a sense of the important factors that together dictate the functionality of proteins. Again, just to summarize very briefly, there are two factors that are absolutely essential in dictating what the function of any given protein is going to be. Factor number one is the shape of the protein, and factor number two is the location in the cell of the protein. The shape is a consequence of the amino acid sequence of the protein, and the location is also a consequence of the primary sequence of the protein, the amino acid sequence of the protein. So therefore, the amino acid sequence of a protein plays a key role in dictating the actual function of the protein in the cell. And with that, we have finished this very brief video, which introduces you to the idea of protein function and the elements that actually dictate that functionality of proteins in the cell. If you like this video, indicate it by clicking somewhere along this line and subscribe to my channel. I will be making a real effort to post videos more frequently, so activate your notifications as well. Thanks so much for watching. Take good care.